Hi guys, welcome back to the channel. Again, we're discussing in this video about restriction endonuclease enzymes. So we'll start from where we left in the last video. So let's go. So this was the point we left at the last video. So these were some of the types of restriction endonuclease enzymes based on the structure, recognition sequence, and nature of cut restriction endonuclease enzymes, which are classified in three major types, which are type 1, type 2, and type 3 restriction endonuclease enzymes. Alright, so I'll be discussing about all of the types and how are they useful. So let's move on. So talking about type 1 restriction endonuclease, these are capable of both restriction as well as modification activities. Alright, the cofactors are S-adenosyl methionine, ATP, and Mg2 plus are required for their full activity. Alright, so these are capable for restriction as well as modification activities. Alright, and also these have cofactors such as methionine, ATP, and magnesium. So, and these contain some of the important stuff that you need to remember, that these have two R subunits which are involved for restriction purposes. It has two M which are involved for modification. These have two M subunits which are involved for modification, and these have one S subunit which is involved for specificity subunit. Alright, so these are some of the important points, and also this is a very important point that this cleave DNA at random lengths for recognition site from recognition sites. Yeah, these do not have a specific sequence, whereas these cleave at random lengths in a foreign DNA. Alright, so moving on from there on. So talking about type 2 restriction endonuclease enzymes. So these are most commonly available and used restriction enzymes. Alright, the type 2 enzymes have a number of advantages over type 1 and type 3. Alright, so you can basically say the type 2 is one of the most important among the three restriction endonuclease enzymes. Alright, so let's come into the third point, which says that the restriction and modification are mediated by separate enzymes. Alright, so it is possible to cleave the DNA in the absence of the modification or restriction subunits. Alright, also it contains the R, which is the restriction, and the S subunits only. It does not have M subunit. Alright, so it does not have the modification subunits. Alright, so basically it is mediated by separate enzymes, so it does not matter if it has or not. Alright, also the restriction activities do not require cofactors such as S-adenosyl methionine and ATP. Alright, whereas type 1 has it. Also along with this, type 1 has methionine, ATP, as well as magnesium, whereas it has none. Alright, and the most important type 2 recognized sequences are usually defined as usually symmetrical palindromic sequences and cut variant. Basically that is very misunderstandable when we are talking about restriction endonuclease enzymes, that it recognizes specific inverted palindromic sequences, all right, in a foreign DNA, so that it can recognize that particular sequences and cut within it. All right, so moving on. So these are some of the examples of type 2 restriction endonuclease enzymes. Alright, so the first example is EcoR1. Alright, so EcoR1 strain, which is the first enzyme, and it has recognition sequences like this. Alright, so this have these form sticky ends, as very much well known, and I've already discussed in my video, first or the second video of this chapter. Alright, so coming to BAM-H1. Alright, so this has Bacillus amyloliquefaciens strain. Also this have BGL2, SAU3AI, A1, ALU1, EcoR5, HINF1, PVU2. Alright, and these some of them form blunt ends, some of them form sticky ends. So these are some of the things that may come in your MCQ questions, that which of the enzymes, or which whether ALU1 or HIN35 form blunt ends or sticky ends. Alright, so just remember which recognition sites, or which of them form sticky ends or blunt ends. Alright, so moving on from there. So Let's talk about the production of sticky ends. I thought that I should include this thing so that it will be like a very good combined uh, viewpoint of understanding what is sticky ends and how does it work. Alright, so as I've mentioned that these restriction endonuclease enzymes recognize inverted palindromic sequences. Alright, so this is the inverted palindromic complementary sequence. Alright, so this you can see, but perfectly separated inverted palindromic sequences, which leads to the formation of sticky ends. Alright, this is eco R1. This is done through eco R1. So eco R1 always forms sticky ends, as well as Hin3 always forms sticky ends, though the sequences are different, but these are complementary palindromic sequences, inverted palindromic sequences. Alright, so this is how this thing production of sticky ends works. So let's moving around. So let's talk about the blunt ends as well. So how the restriction enzyme to enzyme cuts. So the formation of blunt end is like it does not recognize inverted palindromic sequences, but these sequences are definitely complementary to each other. Alright, so they cut in the middle of the sequences, somewhat middle of the sequences. So you can see in this slide that it is exactly in the middle, alright, as shown. So these are complementary to each other but do not recognize inverted. These recognize inverted palindromic sequences, but pardon for my words before. Alright, let me just clear once more that these recognize palindromic sequences. Alright, so these recognize palindromic sequences, as you can see. So there is a difference. Alright, so there is a difference in the sequence order and the pattern. Alright, these recognize sequence inverted palindromic sequences but do not cut at this just like eco R1 and Hin3. So obviously eco R1 and Hin3 falls in different category of restriction endonuclease and have different cut sites. Alright, so due to the presence of different cut sites, alright, so they have a different so they cut differently, and this small one has a falls in a different category of restriction endonuclease enzymes, so that this cuts in the middle of the sequences. Alright, and thus it forms blunt ends, alright, and not sticky ends. Yeah, this is just one of the differences, that it forms blunt end and not sticky ends. Okay, ALU is also one another enzyme. Also, yeah, I just provided you the table, so you can just refer on with that table to understand which of them form sticky end, which of them form blunt ends. Alright, but this is a very important point that I like mistakenly told you wrong. Alright, so basically all of them recognize inverted palindromic sequences. This is a very important point. Alright, so all of them recognize inverted palindromic sequences which are complementary to each other, and their cut sites may differ as they fall into different categories. So depending on the category, their cut site differs, and also that is different due to formation of blunt ends and sticky ends accordingly. Alright, so these are some more examples of cleavage patterns of restriction endonuclease 2. Alright, so this is the sticky end, this is the blunt end, this is the sticky end, this is the blunt end. This is how all of it goes. Alright, so as I've explained it before, so let's move on with that. So let's talk about the type 3 restriction endonuclease enzymes. Alright, so these enzymes also contain more than one subunits, and these have two different subunits in which M, which is the modification subunit, is responsible for recognition as well as modification of DNA sequences. Alright, so M is responsible for two functions, which is recognition as well as modification. Alright, and the other subunit R has nuclease action. So nuclease action is the proofreading action to check whether all of them are properly cut or not, or check for any impurities or everything. Alright, so R is the recognition sequence, as we know, has nuclease action. Alright, so taking to the third point, which is it requires ATP as well as adenosine methionine and Mg magnesium ion for enzyme activity. Alright, so it requires cofactors. All of these three cofactors are involved in type 3 restriction endonuclease enzymes. Also, these cleave only one strand. Alright, so these are not like type 1, so which can cut at any point. So basically these are pretty much specific, and these cleave at only one strand, and these two recognition sites are in opposite orientation and necessary to break the DNA duplex. This is a very common point. So these two recognition sites in the opposite orientation so are necessary to break a DNA duplex. All right. So these enzymes recognize and methylate the same DNA sequences but cleave at 20 to 26 base pairs away. Alright. Whereas the type 1 and type 3 are of little value for gene manipulation, whereas type 2 is the most important among the three restriction endonuclease enzymes. So that's it for my video for these types. So I'll be having one more video for restriction endonuclease enzymes after this. So stay tuned for more. Thank you for watching this video.